Hello everyone, my name is Sepideh Mohammadhosseinpour. I'm a PhD student in Dr. Fabricio Medina-Bolivar lab. I want to present my research, which is about using Arachidin-1 and Arachidin-3 as a potential therapeutic agent for triple-negative breast cancer. Why breast cancer is important? This map shows the mortality of breast cancer globally, from highest mortality in the dark red and to lowest in light red, and USA is in the average of breast cancer mortality. And this map shows the percentage of people who were diagnosed with the breast cancer globally, from highest percentage in dark blue and lowest in the light blue, and USA is in the dark blue area. In 2018, statistics shows more than 266,000 people diagnosed with the breast cancer, which means among 8 women in USA, one of them diagnosed with the breast cancer in their lifetime. For treating breast cancer, we need to understand a term that is called receptor. Receptors are protein on surface of the cells, and they are similar to the doors for the cells. In breast cancer, there are 3 main receptors, which they called estrogen receptor, progesterone receptors, and hormonal epidermal growth factor 2 receptor. The main issue with triple negative breast cancer is this type of cancer cells lacks of all these 3 main receptors. Why this is the issue? Because when we want to treat the breast cancer by using chemo-targeted or hormonal therapy, we need these receptors because drug need to enter to the cells through those doors. And triple-negative breast cancer lacks of all these important doors or receptors. In our lab, we are looking for a way to treat breast cancer by using resveratrol and resveratrol derivatives compounds. Resveratrol is a phenolic compound which is extracted from peanut, berries, and grapes. Resveratrol shows high anti-cancer activity, especially on triple-negative breast cancer in vitro studies. But in a clinical study, resveratrol shows liver failure and low bioavailability and high concentration of resveratrol needed to be effective. So, derivatives of resveratrol have a prenyl groups which showed in the red and blue circle. We call them arachidin or prenylated stimulants. Arachidin show potentially higher bioavailability in compared to the resveratrol in vitro study. And also because resveratrol showed anti-cancer activity, we use resveratrol as a control for this study. The hypothesis of this research is prenylated stimulants have higher anti-cancer activity and higher bioavailability in compared to the resveratrol. Arachidin and resveratrol are defense mechanisms for plants. In our lab, we use peanut hair root systems, and we put the peanut hair root system in a stress situation to produce arachidin compounds. Then we extract those compounds by HPLC. After that, we treat 3 different triple-negative cell lines by using arachidin and resveratrol, and then we test those and their functions by using different in vitro assays, like apoptosis assay, flow cytometry, and cytotoxicity assay. Apoptosis assay and cytotoxicity assays results shows the Arachidin-1 and Arachidin-3, which here you can see Arachidin-3 has the higher apoptosis, which is displayed in the red bar, and lower percentage of alive cells by increasing the concentration of Arachidin-3 in the blue bars. And also, Arachidin-1 showed the higher apoptosis and low percentage of alive cells or low IC50 in these bar graphs. They represent the better results in compare with the other arachidins and resveratrol. We also showed a similar result by using flow cytometry, which is displayed in this graph, and Arachidin-3 in yellow bar and Arachidin-1 in green bar both shows the good result, especially Arachidin-3 showed higher apoptosis in early apoptosis in compared to the control and resveratrol. Therefore, As a conclusion, Arachidin-1 and 3 shows higher anti-cancer activity in compared with other arachidin and resveratrol. And for future study, we want to treat the different type of cell lines, 3 triple negative cell lines and 1 epithelial cell line as a control, and test Arachidin-1 and 3 by different in vitro assays to select the best arachidin, and then we use that best arachidin for in vivo assay. Thanks everyone for listening to my presentation. I want to thank to my professor and all my committee member and my lab members.